Good morning. I'm Dr. Rich McEachin. I'm a research fellow at the National Center for Integrative Biomedical Informatics. And that's a bit of a mouthful, but it's a bioinformatics group at the University of Michigan. I'd like to tell you about an analysis that we recently completed and is now published in BioData Mining. And the title is Modeling Gene-by-Environment Interaction in Comorbid Depression with Alcohol Use Disorders via an Integrated Bioinformatics Approach. My co-authors are Ben Keller, Erica Saunders, and Melvin McInnis. The essential elements of this analysis are that there is lots of data available in public repositories, and we would like to make use of that data, optimize the use of it. And I call these things that we know, but we don't necessarily know that we know them. Wikipedia calls data mining the process of sorting through large amounts of data and picking out relevant information. A statistician hears the term data mining and becomes immediately fearful. But in this case, I believe that we've conducted a very simple and careful analysis of available data to address a specific biomedical question that is of interest to us, which is what are the genetic influences on comorbid major depressive disorder, which I'm going to call depression, and alcohol use disorders. So, first question might be, why look at comorbid alcohol use disorders with depression? And the idea is to identify a more homogeneous phenotype, which potentially would reveal a more homogeneous genotype by looking at the intersection of genes that predispose depression and genes that predispose alcohol use disorders. We may find a set of genes that are common to both phenotypes and are a more simple set of genetic variants that will allow us to understand the etiology, then over time move to more complex phenotypes. So we did 5 steps in this analysis, starting with prior evidence of comorbidity in depression and alcohol use disorders, and that gave us the opportunity to hypothesize that there might be some genetic influences on that comorbidity, so I'll explain that to you. Then that analysis led to the hypothesis that there's a genetic interaction that might be in play, which then led us to the potential for a gene-by-environment interaction, and I'll sum it up by showing you a model that incorporates all of these elements. So, in a lot of cases when scientists do genomics research, they start with very large data sets that are SNP association or microarray data or even linkage data, although that's becoming less popular. And in those cases, the genomics is being used to generate hypotheses. And what I'm going to show you today is we're going to turn that around and look to use that data for hypothesis testing after we generate a more sophisticated hypothesis, a more limited hypothesis. So I generated my candidate genes by a simple database query. And in this case, I used the EntreGene database that NCBI provides to us, and my query was simply depression and ethanol and limited it to the human organisms. And from that query I got 3 candidate genes: tumor necrosis factor, which is a signaling molecule that sets off signal transduction pathways within the cell; methylene tetrahydrofolate reductase, which I'm going to call MTHFR, which is an enzyme essential in folate metabolism; and apolipoprotein E, which you probably have heard of in relation to Alzheimer's disease. In this case, the annotation in EntreGene that resulted in apolipoprotein E being included in the results of the query, the input was inferred from electronic annotation. And in the interest of using only the cleanest data In this case, I eliminated apolipoprotein E from the formal part of the analysis and focused only on the genes that are human curated in Entrez gene. So because I'm using an Entrez gene database query, I'd like to compare a little bit the features of this query to what you might find in a whole genome association by SNP analysis. And I looked at a number of different types of qualifications. In the first case, I considered the availability of data, and in whole genome association, to a large extent, you still have to produce your own data. There are some data sets that are becoming available, but generally speaking, you have to create your own, whereas the taxpayer has already provided the data in Entrez gene, and it really has become a rich resource in the last couple of years. As a result, the expense for whole genome association is high, and the expense for database query is essentially free. The sensitivity of your query in both cases is relatively low. The reason that the sensitivity is low in whole genome association is because you look at a relatively limited number of genetic variants. In the case of the Entrez gene search, there is much that is left to be understood about almost all genes, so the sensitivity of your query is relatively low in both cases. Positive predictive value is essentially how believable a positive response is, and in the case of whole genome association, that's low because most of the results that have been published in whole genome association are false positives. In the case of the Entrez gene query, the data that's in the database has been curated, multiple rounds of peer review, and I'll show you. I did the analysis. You'll find that the positive predictive value is very high. Results of the whole genome association are population specific, and generally speaking, what's in Entrez gene is not population specific. Whether you, in the case of whole genome association, you will find specific variants associated with the phenotype, although in many cases you're not looking at the actual disease-causing variants. In the case of the text-based search, you generally will not identify specific variants, although in a lot of cases there is data about the specific variants in the Entrez gene record. In the case of complex diseases, we're looking at multi-gene effects, and unless you're able to perform an interactions analysis, the availability of that type of data for whole genome association is very limited, whereas it's very easy to do in an Entrez gene query or a database query. Same for complex phenotypes like this comorbidity that is essentially both depression and alcohol use disorders. The query is easy to make, whereas the data is not easy to get in whole genome association analysis. So given that we're just making a simple query against a database, the question remains, is this a believable result? And of course you can check and want to check the references in Entrez gene. But another resource that's available for that type of query is to go to PubMed, also an NCBI available database. In this case, I made queries against the PubMed database, starting with depression and ethanol, and